Hi guys. This is the last piece of our body fluids unit. So finally we are talking about the end of this unit, looking at saliva and urine and other body fluids as well, the identification of them, the documentation, and the collection. So we're going to start by first moving me around. Talking about the difference between forensics biology and toxicology. I did get a few emails about what the test will be on, and some of the study guide was not quite covered yet because we're covering it today. So forensics biology is the nexus between the examination of physical evidence that utilizes both cellular and microbiology subspecificalities in determining if the findings are applicable to crime scene investigation and then in the court of law. So it's really taking all of the fields of biology, mostly cellular and microbiology, and then determining whether or not that evidence can be applicable and admissible in court. Whereas toxicology is the study of body fluids, tissues, and organs for the presence of drug or poison. That analysis then can determine the adverse effects of the chemical on all living things. So we hear a lot of times that MEs will run tox reports, but what does that actually mean? Basically, they're just seeing whether or not there was a presence of drug or alcohol on the body and what did that have to do with the crime that happened. Evidence that gets submitted for tox screenings usually are urine, blood, sweat, hair, and saliva. So certain drugs can actually stay in your hair for a long period of time. I'm not 100% sure on the numbers of it, but I do remember reading somewhere once that like marijuana can stay in the hair for up to 2 months. Urine and blood are the two most common sources for law enforcement officers to work with when determining levels of alcohol or drug in impaired drivers and those involved in vehicle fatalities. So urine and blood, often if someone fails a breathalyzer test, they get arrested, obviously, for DUI or OUI, depending on what the substances that they are under the influence of. And they will go in for either a urine or a blood sample as well to determine the actual level of intoxication. Saliva is important to crime scenes because it's an excellent source of DNA. Every time you bite into something, every time you take a sip of something, whether it's through a straw or through a glass, you leave behind a little bit of saliva, which is the best place to pull DNA. It's detected at crime scene at which a suspect may have left evidence, such as a cigarette butt or had a drink from a glass, a can, or a bottle. It can also be pulled off of a victim who has suffered bite marks. And then we also find it in chewing gum, envelopes, and stamps. This pile of cigarettes here could have more than one DNA profile in it. It could show a relationship between the victim and the suspect. It could also tell you how many suspects were actually involved in these crimes. So you wouldn't just want to collect this one here. You want to collect all of them. Most criminals are not super smart. They will see a water bottle on the counter and fill it with water and take a sip of it. They will, they're not smart. They don't think, oh, I can't take a sip of something. I can't smoke a cigarette here. I can't spit out my chewing gum because it leaves behind DNA. They don't realize that. So we use, these cigarette butts or this piece of chewing gum behind. We can analyze envelopes or stamps that have been licked to pull a DNA profile off. And then, of course, our bite marks, which is actually how Ted Bundy ended up getting arrested. When you're collecting saliva, if it's possible, you do want to collect the entire piece of evidence. Obviously, you cannot collect an entire bite mark. You would swab for this. So you take your swab and you don't rub it directly over the bite mark because you don't want to ruin the integrity of the bite mark itself. You actually go on both the outside of the teeth mark and then the inside of the teeth mark to collect the saliva. Because we're going to end up taking an impression of our bite mark later. For evidence, you do want to bag anything containing saliva in plastic. You want to preserve the DNA. You want to preserve moisture. You don't want to let it to dry out. Even if it's already relatively dry, you still want to bag it in either a plastic baggie or a plastic container, depending on the size. Then again, as always, seal your bag and initial your seal, date, give it an evidence number, all that fun jazz. Urine should always be collected by a medical staff member if the suspect is taken into custody. So police officers, secretaries, they cannot handle urine. It has to be handled by anyone in the medical field, so the medical professional themselves, the ME, or one of their support staffs, but they have to be a medical professional. To collect a sample of urine, a search warrant actually usually is required. If someone is arrested for being under the influence while driving, whether it be drugs or alcohol, that gives you probable cause. If they test positive in the field, that gives you probable cause and you won't need a search warrant moving forward. But if you're looking at someone who committed a crime, then yes, you are most likely going to need a search warrant to get a sample of that DNA. Urine is mostly used to test for the presence of drug or other intoxicants, so drugs, alcohol, anything like that, during the time of the crime. Samples then must be refrigerated quickly and delivered to the lab within 2 to 3 days at most for analysis. Most of the times, because they are collected only by medical professionals, they are brought right to the lab and then analyzed in order, but the analysis does have to happen between 2 and 3 days because you can actually break down the toxins in the urine the more that it sits there. There are a lot of benefits to toxicology screenings. This allows investigators to understand the suspect based off of what may have happened in their systems. Drugs play a huge role in the crime world, and each drug has a different effect on people. This is kind of a topic of controversy right now, is the fact that recreational marijuana. In the state of Massachusetts, it is legal over a certain age to have recreational marijuana, and more states are kind of jumping onto this methodology. But that marijuana has a different effect on every single person. It's not always going to mellow you out. For some people, it does the adverse effect, and it actually heightens them, and it makes them more anxious, and it makes them do things that they normally wouldn't do. So all drugs, everything from recreational marijuana to alcohol to, crystal meth and ecstasy, they have a different effect on every single person that they are absorbed by. Every person's body reacts differently to these drugs. So understanding what the person had in their system helps put together a timeline of what behavior was abnormal for that particular person. I'm not going to show you this video, but go ahead, click that link. You guys can watch a toxicology screening happen. They're going to talk about a urinalysis, which is determining if the urea in the urine has evidence of drug or alcohol. There are a bunch of different tools for toxicology. DNA analysis is divided into 4 separate parts. So you start with extraction, so removing the DNA from the sample. So whether it's from blood, a cheek swab, saliva, urine, hair, anything like that, we're extracting the DNA. Then we have to amplify it. We have to make it larger because most DNA samples on a cheek swab or in a piece of hair or in blood are very small, so we need to amplify it. We're replicating it over and over and over again until we get a large enough sample. Detection, we're searching for the DNA that we're looking for, and then interpretation. We're comparing the DNA sample to known or unknown samples. We also have something called polymerase chain reaction, or PCRs. This is your amplification piece. It's responsible for generating multiple copies of DNA sequences, sections of the gene, or entire genes from a single sample of DNA. This is how we are able to determine samples and make them large enough to determine whether or not a known matches an unknown or an unknown matches several knowns. During PCR, the DNA gets denatured, so it's heated. The double helix strand gets separated from one another. Annealed, so the DNA then gets cooled, so a primer sequence can bind to the DNA. This is basically the lab forcing DNA replication to happen. So we have to heat it to separate it. We have to cool it to add the primer, and then extend it. So free-floating nucleotides get added to the strand of DNA to elongate it and make multiple copies of it. We are able, sorry, I have to keep moving myself today. We are able to make a DNA fingerprint from PCR. This uses gel electrophoresis. Anyone who has had CP Bio, you have done this, or General Bio, we did this electronically last year. PCR makes copies of the strand of DNA. So gel electrophoresis is what's responsible for making a DNA fingerprint of the strand of DNA. This is essentially what your DNA fingerprint will look like in the end. And no two people, except identical twins, have the exact same DNA fingerprint. It's a pattern of DNA fragments that is unique to each individual. So looking at this, you can do it 3 different ways. In a tube, and separates out the bands that are weighted based off of how many base pairs they have. So shorter base pairs land here. Larger base pair can obviously run longer. We usually use the horizontal slab in our labs in our school. Most people use the horizontal slab, but I have seen the vertical slabs done as well. The tube gel is pretty rare. Very few people do it now with how quickly these two can run. The application, sorry, I got like really excited with this PowerPoint last year, I guess. The applications for DNA fingerprint, first is solving cases of disputed paternity, so father figure, or maternity, mother figure. So rarely we see cases of maternity because it's kind of hard for a woman to deny whether or not she had a baby. But maternity comes about in adoptions. So people that are going back to look for their birth mother go through maternity. Obviously, Maury, we know you are the father or you are not the father. Those cases of paternity, determining whether or not someone is actually the father of the baby. Then we have criminal identification in forensics. This is the #1 reason that we use as forensic scientists. We are determining whether or not the DNA fingerprint found at the scene matches the suspect. Criminal identification is found in blood, saliva, urine, hair, or skin cells. Pretty much every time you touch something, you leave skin cells behind, which leaves traces of your DNA. We can take these small traces of DNA, whether it's just a few skin cells, some hair, urine, blood, or saliva, anything like that, and run a PCR and a gel electrophoresis to create a DNA fingerprint that is unique to you to compare to known or unknown samples. And lastly, we are able to do personal identification. So in cases of mass terrorism or mass murder like 9/11 or anything like any of the big school shootings that we're unable to identify someone based off of face or fingerprint, we can take samples of their bone or DNA for personal identification. We can create a genetic barcode that is unique to all individuals except identical twins, which kind of leads to a question. Should this actually be put into practice? Should every baby that is born have their DNA fingerprint run? And some people would say, yes, absolutely, let's do it. Then we have it all in one place. And, every time someone passes away, we don't have to fight for a fingerprint or wait for next of kin. We can identify them right away. But then you have the people that are like, wait, hold up. My kid is going to be raised to never get into trouble, to never do this, never do that, blah, 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 blah. And they're totally against it. And I get that, because then, it leads to the question of, as babies are born, they need to be registered, where in third world countries, that doesn't always happen. People have babies at home. People don't get proper birth certificates for them, especially in these third world countries where they don't have to register for school. They just go into town to the village, they go to school, and they come home. There's no real registration of them. So it poses one of those, like, how do you feel questions, one way or the other. Is it a yes, we totally need to do this, or no, I think this isn't a thing? So it kind of leads to that, like, that's a personal opinion. Do you want to do it? Do you not want to do it? It's, what it is. So hopefully the rest of this PowerPoint gave you guys the rest of the information you need for your study guides. Just remember that Friday during class time, we will be reviewing on a Google Meet. So that is already in Google Classroom for you guys, that link. So I will see you guys then. And Tuesday, because I have you here, Tuesday is your test. Your test will be released at 11:30. So just about when class would start. I know class usually starts 11:40. That's fine. 11:30, and it'll be open until 1:15. So just into fourth block for my CTE friends. It will be open during that time frame. At 11:15, the test will be locked. If you guys are working or cannot make that time commitment for whatever reason, please just let me know as soon as possible so I can make arrangements for you to take your test at another time. You will only get that same time frame to take your test. So just plan on Tuesday from about 11:30 to 1:15 to take your test. It will definitely not take that long. It's not a long test, but I want to give you guys plenty of time. And as always, if you guys have any questions, just let me know.